All right, good afternoon, everybody. Okay, so are there again any visiting students still? No, that was just a one-day thing. Okay, thank you. All right, so the last day we were finishing up on our topic of language. By the way, I was quite happy last time when the room was so much darker. I don't know if that applies to you guys too. Do you guys feel like I just had a better experience overall because the room was darker and I could better see my slides and you guys could better see my slides? I don't know. Maybe it was also all those extra students made everything seem so much more engaging. If you could help me roll the blinds down, I think that would be great. Thank you. So we were talking about the end of the topic of language, and we had ended on this idea that sounds of languages around the world are really quite varied, right? So English has about 40 something phonemes, right? So those are sounds that are specific to the English language. But a lot of sounds that we make are actually, that our vocal tracts are capable of making are actually not available in English. So tone language is a big class of languages that use pitches to convey specific meanings, right? And so one example is mainland Mandarin Chinese, right? That has four separate tones, right? So the same syllable, ma, said four separate ways, can give you four different meanings. And so there was that dramatic example where there was a whole story that can just be constructed from just different ways of saying shi. And so I wanted to end the topic of language on a little bit more about bilingualism, or when people speak multiple languages, what happens in their brain, right? So we know that there are sensitive periods or critical periods. They used to be called critical periods, but now it seems to be more politically correct to call them sensitive periods. And this just refers to a window in time in your lives when you're particularly good at learning something. Right? So the reason why it's no longer called the critical period anymore is that these are not an all or none thing, right? So you can have a gradient of sensitivity. So sensitive period seems to be a more appropriate term than critical period. But either way, there are these constraints, right, that our age seems to put upon us in what we can learn. And this is especially obvious in language learning. So we generally think of these as maturational constraints. So that just means that as your brain matures and your cognitive capacities mature, right, over time, as you develop, that development itself places some constraints, right? It constrains what we can easily learn. And so this is especially true of language. So consider if you're an immigrant, right? You're suddenly placed into a new environment where you have to speak a language that you've never been exposed to. Of course, the age of arrival of an immigrant will constrain, right? Will kind of limit how well they can speak the language of the adopted country, right? So this is a kind of classic finding from that relates the age of arrival of different immigrants to the U.S. That and that age of arrival affects how you perform in grammar tests, right? So a person who arrives late in life, let's say after the age of 18, in let's say in the U.S., even if they seem like they pretty much are very fluent English speakers, there might be tests that are sensitive for differences in grammar processing. So, for instance, things like prepositions. So these are very subtle uses of grammar. Might be a little bit less, a little bit worse in people who arrive late in this country. So I happened to arrive to the U.S. or to Canada actually around age 13. So yeah, I think I'm still on the bottom part of that curve. So, but the point of this is that depending on when you start to enter an environment where you're getting a lot of exposure to your second language, that really changes how your brain processes that second language. And so this was explored in this study by Kim and colleagues that really specifically looked at age of acquisition of a second language. So what they did was they compared a few different kinds of bilinguals, either early bilinguals who were exposed to both languages since infancy, or late bilinguals who did not get exposure to their second language until early adulthood, right? So early adulthood, usually age 18 and onwards, right? And there were different kinds of bilinguals. You'll see that in a moment. So their first and second languages were different. What these subjects were required to do in the functional MRI was a sentence generation task. Right? So things like describe events that occurred during a specific period of the day. Right? And then so they had to do these generate these sentences. And what they were interested in is activity that is in response to thinking in both languages. And this is quite a few years ago now, more than 20 years ago. It's kind of remarkable that the Broca's area, right, which we've talked about as in the inferior frontal gyrus, was overlapping during the generation of first and second languages, L1 and L2, for early bilinguals, right, compared to for late bilinguals. So while the early bilinguals used the same part of the inferior frontal gyrus, the same Broca's area, to process their first and second languages, the late bilinguals, in contrast, use a more prototypical central part of the Broca's area for their first language, whereas the second language actually activated a region that's posterior to that, still in the inferior frontal gyrus, but a little bit less kind of central to where the Broca's area activation would have been. And it's as if the late learned second language is kind of needing to find a different area to occupy within the same overall larger brain region, right? So this separation was especially clear for Broca's area. It's not so clear for Wernicke's area, right? So it seems like there are some things about language production that are especially differentiated between your first and second languages in late bilinguals, whereas areas of the brain that are important for sentence comprehension, word recognition, Wernicke's areas, those are more overlapping between your first and second languages. So this really shows us how the sensitive period really affects the brain, but it affects especially some areas of the brain. So it's not just that you're generally worse, but that there are slight differences in your sensitive period over time, and they apply to different parts of language. So I just want to conclude a little bit on our topic on language. So we started by talking about classic areas, which, since the first day of this class, we talked about the Broca's area, or inferior frontal gyrus, and then since then we've learned about Wernicke's and archaeofasciculus. But then the newer models are more dual stream models, so it's pretty much accepted that by now that those aren't the only regions in the language network, but they're really two separate streams. The dorsal stream pretty much includes the articulatory regions, right, so closer to motor areas and beyond, whereas the ventral stream includes areas such as the temporal pole and the middle temporal gyrus, areas that are ventral and going towards the anterior part of the temporal lobe. And the ventral portion is more useful for lexical access. So lexical access versus articulation, right, those are the two streams of the dual stream model. And then we also talked about not just phonology and syntax and semantics, which are kind of the classic components of what the study of language in the brain is all about. But then there are topics like prosody and pragmatics, which we now think of as more right hemispheric functions, in contrast to the kind of older classic story that left hemisphere is specialized for language. We really think of language as a bilateral function, especially things like pragmatics and prosody tend to be more involve the right hemisphere more. And these are obviously, they activate areas beyond the auditory areas. So the auditory pathway kind of feeds into a group of different areas on the around the perisylvian regions of the brain that are involved in language. Are there any general questions about language? Okay. Hmm.
I just have a question about the study on the previous slide.
Yeah.
Did they look at people who like weren't bilingual but had learned like some of the language at an early age? Because I actually learned German when I was like 4 to 6 years old, but I don't really remember much of it.
Right. So in this study they didn't, but since then they've done all kinds of follow-ups of this experiment, and in general, the findings are that as long as you acquire a language relatively early, so let's say like beyond before teenage years, you tend to use the same Broca's area for it. Yeah. So I think that's really quite remarkable. But I think the story is not completely finished yet, because there's also work now on artificial language learning, right? And it looks like you're not just seeing Broca's area separation, but there's some more subcortical areas that you didn't see in this paper because it is from more than 20 years ago. But things like the basal ganglia turn out to be quite important in learning artificial languages. And of course, artificial language is kind of a subset of what you would be doing in natural language anyway, so we think that there are subcortical involvements as well. So that's kind of where more some of the newest stuff is going. So don't let that confuse you for the purpose of tests. But but I think for now, I think it's the most important point I'm trying to make here is that the late versus early bilinguals do differentiate, especially in the Broca's area. Yeah. Okay. So the bulk of today was supposed to be talking about emotion. All right, so what I think will happen is we'll not finish on the topic of emotion today because it's quite a gigantic topic. So we'll flow a little bit into the next day, where we'll finish talking about emotion and then start talking about decision making. And then next week there's this. We'll finish talking about decision making. We'll talk about executive function, and then the following day I think we'll talk about social functioning. And by the way, this is just a heads up. There's the university relations wants to have a photographer here next Thursday to photograph this class. I'm not quite sure, but so just as a heads up, they promise to be as undisruptive as possible. Yeah, so in case there's a photographer here snapping pictures next Thursday, it's not just that I want to be on photos. It's the university people. Okay, so just a heads up there. Okay. Yeah, and I think they'll let us know before if they need to sign anything or whatever. Okay, so let's start talking about emotions. Okay, so for the remaining few lectures, we're really starting to move towards more high-level ways of thinking about the brain. Right? So from a more psychological perspective, what are our experiences, and then how do lab experiments kind of test these experiences, and then how are these experiences implemented in the brain, right? So we'll start by going through the more psychological models of how people think about these topics, and then we'll look into some methods and some lab studies that people have actually done to scrutinize these topics and to test these psychological models, and then we'll look into the brain imaging studies usually that look into how these processes are implemented in the brain. So we'll start by talking about basic emotions and whether they are universal, and then we'll talk about some of the methods, and then we'll talk about some of the anatomical structures of the brain. And this is my little cheat sheet here. Right? So here are basically the most important areas of the brain that are important that are useful for emotions, and it is very much a cheat sheet because when I'm saying limbic system, it's actually a set of different regions that we'll go into in more detail. But the amygdala and the limbic system and the prefrontal cortex together really give rise to our emotions. And so let's start by, from a more psychological perspective, let's talk about basic emotions. Okay, so if you had to think about what emotions are, everyone can think of a few possible emotions in their heads. And these are probably captured here. Right? So you can think about happy and sad as kind of prototypical emotions. You might also think about being scared as one emotion, right? Being angry as another emotion. And so these are the 6 basic emotions that Paul Ekman in his seminal article about universal facial features of emotions talked about. And he thought, Ekman thought that there are basically a few emotions that are truly universal across different cultures. And he came across this by analyzing facial expressions. I saw obviously happy faces. There are no cultures in the world where people's corners of their mouths don't turn up when people are happy. So that seems to be a very universal expression of emotion. And you can have a worried face and a disgust and anger. So in each of these has a very characteristic set of facial muscles that are activated that are correlated with expression of that emotion, right? And so again, these are very cross-culturally similar. And so even in a very different culture, people, when they're happy, they use their the corners of their mouth and their eyes and so on. And so you can think about whether these expressions of emotions really, how they actually relate to the experience of the emotions themselves, right? So is it that when you're smiling, when you feel happy, you smile because you feel happy? Or is it the other way around, right? So this is a kind of a long-standing debate in psychology. So William James and James Lange theory proposed that emotions actually result from physiological changes and not the other way around. So it's because you're smiling that you are happy. And so this is kind of expressed by this very Victorian idea that William James articulated in the 1800s. You refuse to express a passion, it dies. So you can actually use your physiological or your face muscles to kind of control how you feel. And it's not just that you have no control over your emotions. It's something that your physiology can actually mediate your emotion and your emotional experience. So this leads to newer work on that relates the muscles of the face, like the physiology of the face, to emotional feelings. And so classic study on the facial feedback hypothesis looked at this. Maybe we should try this a little bit. So maybe we'll do a between subject design, right? So half the room, you all have pens or pencils available to you. Everybody find a pen or a pencil. And we'll have this half of the room hold the pencil horizontally on your mouth like this. Yeah. And so this forces a kind of smiling facial expression. Right? Whereas everybody on this side of the room, the other condition, hold your pencil on your mouth like this, like that. Right? Okay. So this is forcing a more frowning kind of expression. And so what Strack et al. Did was actually measure emotions or self-reported emotions after people were forced to hold their pencils either that way or that way. And it turns out that people who were holding their pencils horizontally reported more positive feelings afterwards. So this goes back to this idea that I just presented, that it's not just you smile because you're feeling happy, it's that you're feeling happy because you smile. Right? So there's this kind of feedback loop that your emotion, that the physiology kind of imposes on your emotions. And so it's not just the facial features around the mouth, it's also muscles around the eyes. Right? So when we're smiling, we don't just use our mouths, we also use the orbiculus oculi, so these are muscles that are around the eyes, and also the corrugator muscles, which are muscles around your eyebrows. And so the consistent and correlated activity of the corrugator muscles and the orbicularis muscles and the zygomatic muscles, which control your mouth movements, all together give rise to smiles. And so, and a genuine smile, also known as Duchenne's smile. So Duchenne was a physiologist who actually recorded people's muscles while they were making different kinds of facial expressions. And so he coined he said that a genuine smile has to be characterized not just by the zygomatic muscles moving, right, that make the corners of your mouth go up, but also the orbicularis muscles that go around your eyes. Okay, so this is my friend Chris Ovius, who made these images. I actually remember when they did this. He was a grad student at the time. He's now a professor at UC San Diego. But they had to. I think their advisor was writing a textbook, and so they were asked to make real smiles and fake smiles. So for one of these examples, they had to be told jokes until they smiled, and then they had to have a camera ready for that. And for the other example, they were just told to give a polite Pan-American kind of smile. Which one do you think is the real smile? The one on the right, right? And so it seems kind of obvious. I mean, that one is just a more polite-looking, like smile and nod kind of smile, whereas the other is actually being amused. And I think the key difference here is the muscles around the eyes. Right, so one is just the Pan-American, the other is the Duchenne. This is now actually in the textbook. All right. So this is the idea of basic emotions, right? And that there are some strengths to it. One is that it's cross-culturally viable, right? And it's multimodal, so it involves the involvement of multiple muscles. And then these spatial features actually can be studied very rigorously, right? And so there is a whole line of work called FACS coding, where FACS stands for Facial Action Coding System. And so Paul Ekman, right, the one who initially proposed this basic emotions theory, right, that there are a few different emotions, actually then went on to say that there are particular groups of muscles that have to be activated on the face that then can have a very one-to-one matching system with specific types of emotions. Right? So when you're happy, you activate your zygomatic muscles and your orbicularis muscles and so on. So he's then trained a whole lot of students who now kind of have this whole field on FACS coding. In case you're a TV fan, there was a show a couple years ago called Lie to Me. Anyone know this show? Yeah. And so Paul Ekman was actually the main character of the show. What's his name again? I forget. Main character. He's a cop, but he's very trained at FACS coding. Right? So he's very good at analyzing these micro expressions and being able to tell just based on minute facial expression changes and physiological changes that he could notice. What people were actually feeling and whether they were lying and so on. And so Paul Ekman was actually a consultant on this show. But then there are some weaknesses to basic emotions theory. Right? One is, so if you start out by talking about 6 basic emotions, but are there only 6 emotions, right? So if you're feeling contempt, right, is that more like being angry, or is that more like being sad, or is that like both? If you're feeling in awe or inspired, right, is that like some combination of basic emotions, or is that its own thing? So it turns out that Ekman, right, initially proposed 6 different emotions, and then he added a few more, and then then it wasn't clear anymore how many basic emotions there should be, or whether they were really just continuum of like a few simple dimensions. So the contrasting idea, right, of having a few basic emotions is, of course, having a continuum of different dimensions of emotions. And so this is more a second major category of theories for emotions. And this really looks at emotions as a continuum in space. And so the most dominant kind of theory, a class of theories, proposes that emotions can be thought of as valence versus arousal. Right? And valence can be positive or negative. Arousal can be high or low. Right? So you can imagine a positive valence and high arousal situation. Right? Then that would be on the upper right quadrant. Right? So that's quite positive and quite high arousal. Right? So things that are positive are generally happy. Things that are negative valence are generally sadder, whereas arousal is a kind of energy level. Right? You could have a stimulating, very exciting, high arousal stimulus. Right? That's a stimulating situation. Or you could have low arousal, which is more on the calm side. Right? And so you can think about what emotions would fall under each of those quadrants and what facial expressions would fit each of those emotions. Right? So you can have negative valence and high arousal, so anger, for instance, is negative valence and high arousal. Or you could have someone who's kind of serene, or in a situation that's kind of serene, right? A low arousal, positive valence. Right? It's a kind of a Zen-like positive situation. Or you could have just a kind of boring and a situation where you're kind of negative valence and low arousal. And I think one of the strengths of this continuous emotional theory is that you can think of stimuli as occupying not just certain points in space, but being a vector in this space. Right? So a vector is a sort of an energy change, right, that has a direction and can move over time. Right? So a piece of music, for instance, right, you can start kind of in the middle, but then become more positive and become more arousing, and move around in this space. Another major strength of these dimensional models of emotions is that you can place different specific names for emotions as occupying certain points in this space. Right? So a circumplex model is really quite related to this two-dimensional valence arousal model of emotions, where you can think about each point along a circle in this valence arousal space as having its own semantic category. Right? So you can be tired, which is quite close to droopy. You can have afraid, which is quite close to tense. And these all occupy this two-dimensional space. And these findings are borne out by statistical technique called multidimensional scaling, and so which really helps you find a psychological similarity between different categories of stimuli. So if I were to ask you, for instance, well, how similar are bored and gloomy? Right? You might say, well, they're quite similar. How similar are bored versus excited? Right? They're obviously quite different. And so you can take these ratings of similarity, or similarity judgments, and you can throw them into a statistical algorithm that gives you a multidimensional scaling solution. And a very good fit solution, a well-fitting solution of this multidimensional scaling studies on these perceived similarity judgments falls along a two-dimensional space. And so that pretty much places your perceived emotional categories together on axes that can be called valence and arousal. And so our perception of emotional faces can also fall along this valence arousal space. And what you could also do with these valence arousal spaces is you could then generate stimuli that are designed to elicit certain responses, emotional responses. And so a really well-studied set of stimuli are pictures that have certain kinds of affects, such as it's called the International Affective Pictures System. And so I'll just show you, for example, a few groups of pictures that are from that International Affective Picture System. So right here you have some high arousal, positive valence images. Right? Right here you have some high arousal, negative valence images. And you have, right here, down here, you have some low arousal, positive or negative or neutral valence pictures. I think one downside of this IAPS system is that it's actually quite hard to find something that is both high arousal and zero valence. Right? So the two dimensions are not perfectly independent, or it's hard to find stimuli that totally cover that full space. But then there are some problems with this valence arousal theory as well. So a really obvious one is, well, does it really capture all of the variance you have in emotion? Can there be a third dimension? If there's a third dimension, what should that be? Is it something about energy or tension? Is it something about bodily gestures? So I think that's still a very much an open question. So there are some strengths to the valence arousal theory, is that it's making use of a continuous space. There are some weaknesses. Samples are unevenly distributed. Right? So as I showed you, pictures that are designed to elicit certain emotions it's very hard to design pictures that elicit a neutral emotion. Right? And so how many dimensions are there? What are the dimensions and how changeable are they over time? And we know that things are changeable over time because even given the one, a single stimulus, you can actually observe some systematic changes to what seems to be fairly neutral looking or neutral sounding stimuli. So this is a very classic study by Bob Zients in 1963. This is a very unusually spelled name because Zients is the way you're supposed to pronounce it. So what Bob Zients did at Stanford is he used these Chinese ideographs, which are essentially old-fashioned Chinese characters, and he presented them to people who don't speak Chinese, and he presented different characters in different numbers of repetitions. And then he made people guess what those characters meant. And what he found was that the more people have been exposed to certain ideographs, the more they tended to attribute positive meanings to them. And so certain characters that were exposed more times might be labeled as auspicious or happy sounding or happy, whereas characters that were exposed fewer times got associated with less positive or with more negative meanings. And this is also related to a whole line of work on subliminal priming studies. And that involves repeated exposures to different stimuli that then led to subtle changes in behavior. Right? And so Bob Zients' whole argument is that there are attitudinal effects. Right? We develop attitudes to stimuli that we have been exposed to, and so this is what's called a mere exposure effect. Right? Just merely being exposed to certain stimuli will change our attitudes to them. And so that this can even be true when the exposure itself is not perfectly conscious, right? Even if you don't have awareness of what exactly you've been seeing. And so the really kind of striking finding from these subliminal priming studies comes from presenting stimuli to subjects who have had for very, very short periods of time. So we know that it takes more than 60 milliseconds of presenting some visual stimulus for people to be consciously aware of that stimulus. And we know that we can present things very rapidly. So if you consider the classic film clips, right? So you've seen those like old-fashioned movie theaters where they have these big reels of movies, and they just roll the film, right? And that's actually what you perceive as a moving film is actually a large number of small frames, right? That still frames, right? That are presented together at a rapid refresh rate, right? So you can take some of these still frames and actually splice in some still frames that tell you to do certain things. And so a classic study was they had two movies that they made students watch, right, in a movie theater. So one group of students got to watch a regular movie, whereas the other group of students got to watch the same movie, but inserted as still frames within the film reel were frames that said, "Eat popcorn, drink Coke." And of course, people were given as much Coke as they wanted, as much popcorn as they wanted. And of course, the students who got the subliminally presented messages of eat popcorn, drink Coke, drank more Coke and ate more popcorn. So even though they had no conscious awareness of having seen these messages. Okay? So if any of you've seen the Simpsons episode, there's a Simpsons episode where the characters were putting in like join the Navy, like subliminal messaging of join the Navy, and then that led people to join the Navy. I don't know. Nobody knows the Simpsons episode? Okay. Okay. So these subliminal priming studies, I think the main take-home message is that repeated exposure does change your attitude to a stimuli. And so we've done a little bit of this, right, just with musical melodies that were presented repeated a number of times, and it turns out that the more times you listen to a certain melody, the more you actually rate them as being pleasurable and enjoyable. So And these kinds of effects can also be observed and be linked to arousal changes in physiology. Okay, so sometimes you can actually have changes in arousal levels that can be observed physiologically that can then get attributed or misattributed to a different source from where it came from. So this is a classic example. It comes from, it's called the Capilano Bridge experiment. Has anyone been to the Capilano Bridge? Where is Capilano Bridge?
It's from Vancouver.
Yes, right. So it's just north of Vancouver, where I'm from. So North Vancouver. And it's a very beautiful, I should actually replace this with, I was there recently. It's a very beautiful dangling bridge over very steep Capilano River. And so when you're walking across the bridge, it sways a little bit. And so that causes your heart rate to rise a little bit, and it causes your skin conductance, or it causes you to get a little bit sweatier. And so these physiological changes just happen as a result of being on this slightly dangerous situation. And so what Dustin and Aaron, who were professors at the University of British Columbia, which is in Vancouver, what they did was actually take advantage of this Capilano Bridge to design an experiment to look at how these physiological changes might be misattributed or easily attributed to a different source of stimulus that gave rise to that emotion. And so I'll describe this below. So in addition to Capilano Bridge, it turns out that just a mile or so downhill, down the river, there's another bridge that also crosses the river, but it doesn't sway like the Capilano Bridge does. Instead, it just has this concrete, not very exciting bridge. That's actually a pretty well-matched control for the Capilano Bridge. So what these researchers did is they had a very attractive female research assistant. It's like very PC, okay? So it's an attractive female research assistant, and all the study participants were males. And the research assistant would take each of these male participants, go across either the Capilano Bridge or the control bridge, and then on the other side of the bridge, they would sit down, and the participant was given the task of writing down some scenic descriptions of the environment. Okay? And then they walk back together the way they came. And then at the end of the experiment, quite crucially, the female research assistant goes, "Oh, that was fun. If you have any questions about the experiment, this is my phone number." The dependent variable, so what the researchers were actually measuring, was how many male research subjects actually called the female research assistant to ask them out on a date. And it turns out that there were more subjects who did call the research assistants after they'd been on the Capilano Bridge compared to the concrete, not so arousing bridge. So the interpretation is that when you're crossing this scenic and slightly vertical inducing situation, that causes physiological changes in your arousal levels, and that's actually those changes then get misattributed to the female research subject, right? So essentially, the males are saying, "Oh, like my heart's beating so fast, and I'm getting so sweaty. I must be in love with this research assistant." So effects of the environment can actually cause arousal changes that then lead to the misattribution of emotional experiences. So I think that's really interesting to think about. And because of this study and a number of other studies, these psychophysiological methods still end up being extremely useful for how we think about emotions. So psychophysiological methods just refer to physiological changes, right? So changes that can be measured in your biology that are related to psychological processes. Right? And so what does a psychophysiology lab have? And so things like measuring sensors for skin conductance. Right? So Sean's thesis, which is about to be filed tomorrow, actually includes skin conductance measures. Right? So this is a pretty classic setup. Right? So BioPack is one of these companies that makes these devices that help you measure how sweaty people are. And the sensors themselves can be just placed on the surface of the fingers. Then you can also record respiration, so the breathing rate, and those can be recorded just by attaching electrodes to the rib cage. So these are a few possible kinds of measures that we can use. So blood pressure and ECG, so electrocardiography, so heart rate variability, electrodermal activity. So essentially that's the same as skin conductance, right? So if we're aroused, then we sweat a little more. So skin conductance, electrodermal activity is a pretty big one. EMG, so muscle activity, electromyography, that's what EMG stands for. So muscle activity, EEG, you can also have an EEG. This is actually the EEG. So you can actually have EEG recordings that are part of these devices. And then also eye movement, right? If you're more aroused, you might move your eyes a little bit more. So electrooculograms can be part of the setup. And then finally respiration. So I'm not going to ask you to be able to recognize these different kinds of traces, but you do have to know the different sources, different types of psychophysiological recordings. And so these different psychophysiological recordings together give you a pretty good sense of the major components of the nervous system that are important during the perception and experience of emotions. And those include, there are two major kind of complementary pathways in your nervous system, one of which is the sympathetic nervous system, which is responsible for your flight or fight responses. And so when you're highly aroused, right, your pupils might dilate, right, and then your sweat glands might produce more sweat. And then your hairs might stand up more, right? So that's the ancient kind of response to help your body be prepared for fight. Right? So that's the sympathetic nervous system. In contrast, you have the parasympathetic nervous system, which gives rise to your rest and digest response. Right? So when you're resting and digesting and knowing that there's nothing particularly dangerous around you, you might constrict your pupils more. Right? You might start to activate your digestive system a little bit more and activate your intestines and your stomach more. So those that kind of interplay between the parasympathetic and the sympathetic nervous system, that's the reason why we look at different kinds of responses within the skin conductance signal and also from the heart rate. Right? So both parasympathetic and sympathetic nervous systems control heart rate. Right? Okay. So the purpose of these psychophysiological recordings is to give us a window into the ratio, right, of the sympathetic nervous response and the parasympathetic nervous response. Okay. So let's move on to neural structures and how they are related to our emotional experiences. So from the 19th century onwards, it's been known that there's this major set of brain regions together known as the limbic system that are responsible for emotions. And so limbic means kind of ring. And so they form a ring. These regions together form a ring-like structure around the thalamus. Right? So the thalamus is right there. Right? So together, the classic areas of the limbic system, also known as the Pepez circuit, that includes the anterior portion of the thalamus, right, and then hypothalamus, which is sitting right on top of the thalamus, and then that goes. It also includes the hippocampus and the cingulate cortex. Right? So together, these areas are forming a ring around the thalamus. So the textbook also represents this. I think it's quite a nice diagram of the limbic system. Right? So again, that includes the cingulate gyrus and also the hippocampus, and then it terminates in the amygdala. And then also in the anterior portion of the thalamus. So this anterior portion of the thalamus just corresponds to this part in the previous image. So these areas together are the classic limbic system. And so let's just start by talking about the hippocampus and the amygdala complex. Right? So hippocampus is this kind of long structure that runs throughout your medial temporal lobe, right? And then at the anterior tip of the hippocampus is the amygdala, which has for a long time been known to be a major seat of emotion. Right? If you think about one area of the brain that is important for emotion, most people would say amygdala. But when you look more carefully at the types of emotions that tend to activate the amygdala, they usually pertain to negative emotions, especially to fear. So if you look at meta-analysis of large number of functional MRI studies in the past 20 years or so, the word fearful tends to be most closely associated with activity from the amygdala. Right, so this is from the Neurosynth, right, the meta-analysis engine that you guys did your first project or first homework assignment on. So if you just type in fearful, right, so the bulk of the activity is centered around amygdala. And so once I actually encountered a person with bilateral amygdala lesions. This was a patient, actually, sorry, not a patient, normal subject came into my lab for a normal research experiment. They were just answering an ad for paid subjects, and they came in, and then we realized quite soon after meeting this person that she was a little bit odd, and this is before we saw her brain. She showed her tests were normal, for IQ tests were normal, language tests were normal, but she would just keep asking questions like, "Hey, do you have any food for me?" And she would say things that were a little bit sort of sexually inappropriate, and also crack jokes that were kind of not funny. And we didn't really know what to make of it until my research assistant processed her T1 image and said, "It's like, is this normal?" I'm like, no, this is not normal. So this is the subject's brain, and you can see how the hippocampus, posterior hippocampus is there, but anterior hippocampus is gone, and bilateral amygdala are not existing. So amazingly, this subject has been walking around, living a life, and not really been seeing neurologists or anything like that. But any guess what he does for a living? Besides participate in paid studies? Lawyer? Yes. Oh, that's a good one. Yeah, I think she would probably make quite a good stunt double. Yeah.
Skydiving instructor.
Yeah. So so a lot of fearless. She's actually a stand-up comedian. Actually, not a very successful stand-up comedian. I mean, and I think it really it's very hard to be up there telling jokes and have people not risk having people not laugh at your jokes. So she's actually a stand-up comedian. Have bilateral amygdala lesions. But was making money by participating in studies on the side. So this is obviously an incidental finding. It's quite rare in cognitive neuroscience, but there are more systematic studies of people with lesions, looking at lesion studies. So you can look at large populations, large group of subjects that have undergone brain surgery, or traumatic brain injury, or tumors, or strokes, or any number of brain trauma that could lead to a lesion in particular areas of the brain. Especially, let's say, looking at amygdalas, people with lesions in the amygdala actually show impaired recognition of fearful faces and music. So this is one of the examples of eye tracking that was done on people with amygdala lesions compared to controls. So in this study, people were asked to look at the faces that had certain emotions shown on them, and they were interested in where exactly the subjects were looking when seeing these faces, and then also on whether they could perceive or experience the emotions that were in the faces. And so, first of all, subjects generally did not recognize the fear, did not recognize that these faces were fearful faces. And furthermore, these here are looking at eye tracking data. So these lines are saccades, and the dots are where the fixation points. So these show the dots and lines show where the subjects were looking within the face. And you can see how controls were quite focused over the eyes of the face, and also somewhat over the mouth, but the amygdala lesion patients were not doing that kind of eye scanning at all. We could also play people different pieces of music that were composed to be scary or happy or sad. Right? So this is one example. Let me just do that again. All right, so would you say that was a happy or sad or angry or scary piece of music? Scary sounding, right? So that's quite clear, right? So people with no amygdala lesions have no trouble identifying these composed pieces of music as trying to convey a certain emotion. But of course, people with amygdala lesions were not able to do that. Then there's also some classic lab experiments on fear conditioning. And so these are things that are quite well replicated in animals, such as in mice. Right? So you can have fear conditioning paradigm. So this is based on the classical conditioning paradigms, where you can pair certain stimulus with certain responses. Right? So for instance, an unconditioned stimulus always gives rise to an unconditioned response. So, for instance, if you shock a mouse, right, while it's inside a Skinner box, right, if you give a mouse a shock, they will freeze. Right? So, the unconditioned stimulus of shock gives rise to the unconditioned response of freezing. Now you can pair another stimulus, which becomes known as the conditioned stimulus, with the unconditioned stimulus. All right, so that's adding the conditioned stimulus to the unconditioned stimulus, and that now gives rise to still the response of freezing, but that now becomes the conditioned response. Right, so if you pair a tone, for instance, as the conditioned stimulus, if you pair that with the electrical shock, right, then that gives rise to the unconditioned response of freezing, which has now become the conditioned response. So after this training happened for a few trials, now if you just give a conditioned stimulus or just give the tone, that can still give rise to freezing, right? Even if it's not presented with the unconditioned stimulus of shock. Right? So after a few trials of pairing the tone with the shock, even if you just present the tone itself, just the conditioned stimulus, you still get the conditioned response, right, of freezing. So that's a very well replicated paradigm, and that's a fear conditioning paradigm. Right, so essentially, you've conditioned the rats or mice to freeze in response to something that's originally fairly innocuous seeming conditioned stimulus. So that's the classic fear conditioning paradigm. And what you could do over time is see whether you can disassociate the relationship between the conditioned stimulus and the conditioned response. And so this is the process of pure extinction. So all that involves is just presenting the tone by itself a number of times without the unconditioned stimulus, without the shock. So that means after the rat has learned to freeze to the tone, you just present the tone by itself without the shock. And so a normal, healthy mouse would, over time, forget this fear conditioned response and just end up not freezing anymore. So this is the process of fear extinction. And so you're essentially extinguishing the learned association between the conditioned stimulus and the fear response. Right? So this fear extinction paradigm, it turns out that there are some specific populations that seem to have trouble with fear extinction. And so people with PTSD, for instance, it turns out that when after they've consolidated their fear response, it fails to be extinguished. Right? So this can lead to certain triggers, right, that can induce certain fear responses. So if you see something that reminds you of the experience you had on the front lines, for instance, or in the traumatic situation, it doesn't matter how many times you see that stimulus without the traumatic situation, you still get that unconditioned, that conditioned response of fear. Right. So PTSD seems to be a situation when your consolidated fear response is failing to be extinguished. And I think this is especially important for veterans, right? So soldiers coming back from the front lines. Let's say you take 100 soldiers who come back from a traumatic situation. A lot of them bounce back and stop associating stimuli with fearful responses, but some don't. And those then go on to develop PTSD. So what is it that differentiates the people who later develop PTSD from people who don't? And so what Morey and colleagues found, so they were looking at a large number of patients who were coming back from front lines. And so some of them had PTSD, some of them also had been exposed to trauma but did not have PTSD. And what they found was that people with PTSD have a smaller volume of amygdala as well as hippocampus. So it seems like there are individual differences in amygdala volume that can predict whether people given the same trauma would end up developing PTSD. And this seems to be related to the ability to of learning fear extinction. And we also know that in addition to the amygdala, right, the amygdala is just one of several regions that are important for emotion, and these are generally anatomically connected, close to the insula, which is part of this gray matter structure that's subcortical. So in addition to basal ganglia, which we talked about last time, the amygdala is also quite structurally close to this whole set of areas together known as the insula. And that's also a relatively older part of the cortex. And so that's why it's called archicortex. So in contrast to regular other cortical areas, which have 6 layers, the insula has fewer cortical layers. But what the insula tends to be useful for is the integration of external sensations and internally generated responses to those sensations. And so and it's quite a large area that goes that kind of stretches from the temporal lobe all the way to the anterior temporal lobe and to the frontal lobe as well. And if you look at the anterior to posterior axis of the insula, you can generally think of the anterior insula as like the anterior portion of that, right? Whereas the posterior insula kind of closer to the temporal lobe. And the anterior insula has connections to several regions throughout the frontal lobe, which we'll talk about in a moment. And that's generally useful for coding for arousal and changes in heart rate. And so this relationship, right, that's mediated by the insula, this relationship between what we perceive in the outside world and how we internally generate our physiological responses, that's a very important relationship in regulating our emotions, and the insula is especially useful for that. So I'll, in the remaining part of today, I'll talk about the frontal lobe. Right? So we've talked about the amygdala and kind of moved on to a little bit about the insula. And the last part of today is going to be all about the frontal lobe. And so especially the frontal lobe has a few different major divisions. One is the orbital frontal cortex, which is, for the purposes of this course, you can also think of that as the ventral medial prefrontal cortex. So it just depends on how you look at the brain. And so orbital, so this whole thing that's colored in any color at all, that's frontal lobe, especially in the prefrontal cortex. And so that includes the orbital frontal areas and the dorsolateral and ventrolateral areas. And so especially the orbital frontal cortex, if you take the whole brain and you flip it over and you look at it from the ventral view, right, the bottom part of that, that's the orbital frontal. And orbital just means it's closer to the eyes, right? So you know that the eyes are kind of right underneath there, which means it's kind of closer to us from there. Right? And so if you look at it from the ventral portion, it's the orbital frontal cortex. Some people also think of it as the ventral medial prefrontal cortex, and that really just means that it's closer to the ventral part of the prefrontal cortex, and it's mesial as opposed to lateral. And so think of OFC, orbital frontal cortex, and VMPFC, ventral medial prefrontal cortex, is kind of the same region. In contrast, you have the more lateral portions of the prefrontal cortex, and that includes a more dorsolateral portion of the prefrontal cortex and a more ventrolateral portion. And then from the mesial view, if you look at just the medial prefrontal cortex, there's also a more dorsal view, dorsal part of the medial prefrontal cortex, and that includes the cingulate. Especially the cingulate cortex is the whole gyrus that includes everything above the corpus callosum, which is right here, and all the way from the anterior tip to the posterior tip. But the anterior portion of the cingulate cortex, ACC, anterior cingulate cortex, is really just within the prefrontal cortex, the areas that are within the cingulate gyrus. So this is just another anatomical division of the prefrontal cortex. Right? So in red, I'm showing just the motor cortex. Right? Everything anterior of the motor cortex is the prefrontal cortex, and so the green stuff is generally the dorsolateral prefrontal cortex, whereas the dark blue here is the orbital frontal cortex. And so this differentiation, right, is just another view of the same thing. Right? So we have motor cortex, premotor cortex, and then lateral versus orbital frontal, also known as ventral medial prefrontal cortex. And then anterior cingulate cortex is what you can see from the midline. And then there's the Broca's area, which is part of the inferior frontal gyrus. And then the insula, which is more mesial to that. Okay, so that's a lot of anatomy. But the point is, what happens when you have damage in some of these areas? Well, it turns out that different kinds of damage results in, within the frontal lobe result in different kinds of impairments. And that's why it's particularly important to have a better sense of what exactly we're talking about when we're talking about prefrontal cortex. So what's most striking is that if you have orbital frontal damage, you tend to show problems with emotional regulation, so emotional dysregulation. You tend to show disturbed social behavior, so inability to form normal social connections. You tend to have troubles with decision making and impulsivity and goal-directed behavior. We'll talk about this later on in this course, but you also tend to show problems theory of mind. So theory of mind refers to the ability of understanding other people's thoughts and emotions. And so people with orbital frontal damage don't tend to be able to understand other people's thoughts and emotions. And they also don't tend to show the extinguishing response. So orienting response, which is a very kind of low level response to just general sensory stimulus. Right? And so this can be tested by, for instance, making a loud banging noise to a certain side of your visual field, right? And then just looking at whether you turn your head in response to that. It turns out that people with orbitofrontal damage are much more likely to show an increased orienting response. But this is in contrast to normal fear conditioning responses. So it seems like, taken together, the orbitofrontal cortex is very important for cognitive control, right, and for emotional emotional decision making. So, who's the most classic example of people with orbitofrontal damage? Phineas Gage, of course. I think I showed you a little bit about Phineas Gage on the very first day of this class. So, Phineas Gage was described. It was a case that was described by John Harlow in the 1900s, and so the famous case happened when he was about 24. He was a railroad foreman, and one time he got into a railroad accident where a tamping iron, which is this large piece of metal that you use to tamp down the dynamite that's used to blast railroads when they're working on railroad tracks. So the accident happened when the tamping iron went through his skull, through his brain, and out the opposite side of his head, essentially taking out a chunk of the orbitofrontal cortex. And so this is how Harlow wrote about Phineas Gage after the accident. So he miraculously survived the accident, but afterwards his behavior changed a lot. Right? And so he was described as a fairly nice, sociable person before the brain damage happened. But afterwards, this is what Harlow writes. He is fitful, irreverent, indulging at times in the grossest profanity, which was not previously his custom, manifesting but little deference for his fellows, impatient of restraint or advice when it conflicts with his desires, at times pertinaciously obstinate, yet capricious and vacillating, devising many plans of future operations, which are sooner arranged than that are abandoned in turn for others appearing more feasible. In this regard, his mind was radically changed, so decidedly that his friends and acquaintances said he was no longer Gage. So very clearly, he's showing problems with impulse control. Right? So cognitive control seems to be something that orbitofrontal cortex is especially important for. And we talk about Phineas Gage like he's this very rare and classic case, but of course it turns out that orbitofrontal cortex damage is one of the more common forms of brain damage. So this is a study that looks at lesion overlap, kind of very early type of lesion overlap of many cases that were that had undergone different kinds of traumatic brain injury that were examined post-mortem. And what the researchers found was that when you look at the overlap of many different types of lesions that people had, they tend to cluster over a few different areas, especially in the orbitofrontal cortex, and also somewhat in the anterior portions of the temporal lobe. And so why is that, right? Why might you be more susceptible to brain damage in the orbitofrontal cortex and in the anterior temporal lobe? In 1937, there was a very simple and clever experiment done called the shaken jello mold study. What they did was they took human skulls and used them as a jello mold. So you know how to make jello, right? You have this jello powder, right? And you put it in hot water and then you let it cool down, right? And so that you let the jello set. And so they used the human skull as the jello mold. And then after the jello had set, they shook the human skulls. And what they found was that when they opened up the human skull again to look at the jello, what they found was that most of the damage in the jello was concentrated over areas that would include orbital frontal cortex and anterior temporal lobe. And so the reason for this is that the inside edges, ridges of bones within the skull are sharper in areas that were close to the orbitofrontal cortex and the temporal pole, and that meant that if you hit your head, right, if you have a concussion, you're more likely to damage these anterior parts of the brain. And so very good example of these are coup contra coup concussions. So, so you can have a direct concussion, which involves a direct hit, blow to the front of the head. And so it's not so surprising to imagine how that could actually result in orbitofrontal damage. But you could also have contra coup concussions, which are damage to the back of the head, where the blow actually happens on the back of your head. But what happens there is that the brain could actually jiggle forward in the same way that a shaken jello mold study would have happened with jello in the shaken jello mold study, and this results in the anterior portion of what would have been the orbitofrontal cortex actually being damaged by the bony ridges on the inside of the skull. Right? So even though the blow is to the back of the head, you can actually result in brain damage in the front of the head. So that's what a contra coup concussion is. Traumatic brain injury can result in brain damage in the orbitofrontal cortex, but you can also get them from a number of other sources. So meningioma or tumors, metastatic tumors, or strokes. Right? So those are all possible sources of frontal lobe damage. And so I studied with one of these neurologists, Bob Knight. That's Bob right there. There he is interviewing a orbitofrontal cortex patient and just talking about why he couldn't hold down a job and things like that. And so this is one letter that Dr. Knight got from a concerned parent of a patient. I'll just let you guys read this. Okay, so little did we know what lay ahead. So it seems like she lost her sense of smell, some loss of vision in her left eye, and otherwise she seemed to be fine. However, this is what happened afterward. Promiscuity, daredevil antics, disappearances. So I think it's quite striking that a person who seemed relatively normal after brain damage in the orbitofrontal cortex could have a very dramatic change in behavior. So these are, as I said, kind of general impulse control problems, right? Lying, shocking behavior, promiscuity. So people who have trouble controlling their impulses, I mean, that's quite common among people with orbitofrontal cortex damage. And so in this interview, I don't have a recording of this interview anymore. Dr. Knight asked the patient, why can't you hold down a job? Well, I stole from my first employer, and then I didn't show up to work, and a bunch of behavioral problems. And at the end of the interview, Dr. Knight asked, "Do you think that was the right thing to do?" He said, "No, I don't. I know it's not right. I just couldn't help myself." So it seems like there's not a problem with morally knowing what the right thing is to do, but it's a problem with implementing what you know to be right. And so that's again an impulse control problem. And this is actually, if you're very interested in this work on ethics and how brain damage might change your ethical behavior, there's a course on neuroethics that Professor Nihal de Lannerol teaches on some semesters, so you might find that to be an interesting course. I think it's fascinating. So people with frontotemporal dementia, right, also tend to have impaired metabolism in the orbitofrontal cortex and the anterior temporal lobe. And so generally they tend to show sociopathy, so a lot of problems with social functioning, especially problems with emotional concern for the consequences of their own actions. And this is tied to disinhibition. So this quite an interesting study by Damasio, Antonio Damasio, whose work we'll talk about much more in the next few minutes. What Damasio's group did was they compared patients with different kinds of brain damage who either had sociopathic tendencies, right, and they can devise various neuropsych tests to tease apart people with sociopathic tendencies from people who don't. So when they contrasted subjects with brain damage who had sociopathy versus people who did not have sociopathy, patients with acquired sociopathy tended to have damage over the ventromedial prefrontal cortex, right? So whereas patients with more perisylvian damage, right, so including the language areas that we talked about last time, they tend to not have sociopathy. So it seems like, again, this orbital frontal cortex is especially important for this concern, this social concern for other people's emotions and well-being. And so what Damasio and colleagues did, and I think we don't actually have time to go over this, but we'll start with that next time. What they then went on to show was that the ventromedial prefrontal cortex is actually doing the storage, or is actually storing the links between our actions and our sensory consequences for those actions. And so if, so let's say if I'm a normal behaving human being with a normal orbital cortex, and if I somehow hit you, right? I don't know why I would do that, but if that happens, you would feel bad, and I would feel bad that I hit you. And that would lead to my changes in physiological arousal, right? So I would, my heart rate would have gone up, my skin conductance would have gone up, right? And so all of those relationships, right, between my action of hitting you and my heart rate and my skin conductance going up, right, all those relationships get stored, right? And so that's a relationship between the somatic experience of those physiological changes and my knowledge and my marker of those being related to me feeling terrible, right? And you feeling terrible. So that somatic marker is what Damasio and colleagues who did this research think that the orbitofrontal cortex is actually storing. So the relationship between our physiological consequences of our actions and our actions themselves. So I think I'll stop there, and the next time I'll start with this slide about acquired sociopathy, and then I'll show you some specific examples of that done in the lab. Thank you.